Naika, thank you, Professor Pradeep Divakar. Thank you for joining in today. Ladies and gentlemen, good morning. I'm Beleshanai, the editor-in-chief of Mycoish, Journal of Modern Mycology. Today, we have AFB ILS talk by Professor Pradeep Divakar. AFB stands for Association of Fungal Biologists. I am a member of the managing committee of the Association of Fungal Biologists. ILS stands for Indian Lichenological Society. And we have with us Dr. Sanjeev Naika representing the ILS. Before we hand over the platform to the speaker, Professor Pradeep Divakar, we would take probably 3 to 4 minutes talking about AFB and talking about ILS, and I will take a couple of minutes to introduce the speaker to today's audience. As I told you, today's speaker is Professor Pradeep Divakar from the Complutense University of Madrid, Spain. He'll be talking about lichen biodiversity estimation in the 21st century. Thank you, Professor Divakar, for agreeing to give this talk. About the AFB, we started this association a couple of years back. To tell you, this is one of the fast-growing mycological associations in India. In last 2 years, we have got 250 life members, and we have conducted many online and offline events. And we have conducted a couple of annual conferences. Last was at Pune. Before that, it was at Mumbai. The next one will be at Kolhapur in December 2025. So this is about the AFB. I now request Dr. Sanjeev Naika to say a few words about the ILS. Professor Naika.
Thank you, Dr. Rashanai. I am representing ILS. Of course, among the audience, we have Dr. Gaurav Mishra, who is the secretary of the society. But I'll be telling you about the ILS. So we established our society in the year 2014. Although we started as a lichenology society, but soon we grew into accommodating the interest of other cryptograms also. So now our society promotes not only lichenology but also other cryptogamic groups, and we are 10-year-old and we are organized already 5 mega events. We organize conference every alternate years, and we also keep organizing workshops and training programs. We have about 250 life members so far, and the association also comes out with the biannual journal, that is the Cryptogram Biodiversity and Assessment, and I request you all to contribute your papers into that. And again, as Dr. Shanai told, ours is also a growing society, and we look forward to for the cooperation with you all for the growing further. Thank you very much.
Thank you, Dr. Sanjeev Naika, for those words, and thank you very much for collaborating with the Association of Fungal Biologists. Dr. Deshmukh has conveyed the best wishes for today's event, as he's away from Mumbai, from his residence. Before I hand over this platform to the speaker, let me give a brief introduction about the speaker. Today's speaker, Dr. Pradeep Divakar, Professor Pradeep Divakar. Professor Kumar Divakar is currently working as Professor of Botany in the Department of Pharmacology, Pharmacognosy and Botany at the Complutense University of Madrid, Spain. Please bear with my poor pronunciation of some of the names here. Dr. Divakar has received many prestigious fellowships, including Juan de la Cierva Fellow, Minister of Education of Science, Spain in 2004, Ramon Cajal Fellow, Ministry of Science and Innovation, Spain, Fulbright Scholar, U.S. Department of State, our State's Bureau of Educational and Cultural Affairs, and the Council of International Exchange of Scholars in 2015. Professor Divakar has published more than 160 research papers till now, most of which are published in esteemed journals, including Fungal Diversity, Scientific Reports, Persoonia, PLoS ONE, to name a few. In the past 2 years, Professor Divakar has completed 2 projects as a project investigator and co-PI, and he is currently heading 2 other projects. Dr. Divakar is one of the grant reviewers and panel members of agencies like European Commission Research Executive Agency EU, Yes, Estonian Science Foundation, a national agency for evaluation and foresight, Spain, Czech Science Foundation, Austrian Science Fund, National Science Center, Poland, German Research Foundation. Professor Divakar is also many national and international societies like International Association of Lichenologists, Society of Systematic Biologists, American Bryological and Lichenological Society, Association of Plant Taxonomy, and our own Indian Lichenological Society. Professor Divakar is serving as associate editor for many international journals, including Biodiversity and Conservation, Journal of Fungi, Frontiers in Microbiology, MycoKeys, and he's a guest editor for journals like Fungal Biology and Lichenologist. Professor Divakar has successfully supervised or co-supervised 4 students for their PhD team. With this brief introduction of the speaker, I request Professor Pradeep Divakar to start his presentation. Over to you, Professor Pradeep Divakar.
Thank you, Dr. Vale, for your kind introduction. So, and I thanks also to Dr. Sanjeev Naika for kind invitation for giving the talk today to this wonderful event. So I'll try to share my slides, but it's saying I don't have the permission to show the slides. I don't know.
Give me a second. I will fix that, please.
Yes.
Now you can share the slides, please.
Yes. Yeah. Now I can do it. Yeah. Okay. Oh, it's again.
We can see the slides coming up here.
Okay, I can now. I will open the slides.
You can go to a desktop, yeah, please.
Okay, I will go to desktop now.
Open the slides.
Okay, here we go.
Yes. We can see the first slide. Can you just move to the second slide just for technical testing, please?
Yes.
Yes. This other slide is moving. Please go ahead with the presentation. Thank you very much.
Okay. So as I was telling earlier, it's my great pleasure to be here to this event, and I really thanks to all the audience to joining today. Today is Sunday, so Sunday is holiday, so taking their time to listen my talk. I really thankful to all. So what I thought today, what I can say to both the members of the society and the interest of the mycologist and the lichenologist in general. So I choose a topic that how we can estimate the lichen biodiversity in the 21st century. So this was my idea, and to show you some of other tools that has widely used now and we are using to estimate the lichen biodiversity. I will tell you why it's necessary and to use the tools, not others, and why these tools are really useful to estimate lichen biodiversity. So as you all know, the lichens are an ecologically obligate stable mutualist between a fungal partner that we call the microbiota and the unicellular or filamentous algal that are the cyanobacterial cells. Could be the green algae, and there are also the cyanobacteria, and they form the this is the general a definition of the lichen that we all know. I don't want to go into deep, so I have shown you here a picture showing this is the cortical layer. You could see here the algal cells are merged in and the hyphal cells are there. So it is evident that they form the association between these two organisms. But however, the recent investigation has shown that the lichens has not include only just the two organisms, but the lichen also forms the association with other fungi and also non-photosynthetic bacteria. Because of that, we now say the lichens are holobiont. Holobiont means that they do not include just two bionts, but several organisms are living inside the lichen thallus. In a study that we did in 2020, we found that apart from the lecneromycete, that is the main fungal partner for the lichen symbiosis, there are also other lineages I have shown here. I don't want to go in deep, but we could say there are also other lineages belonging to the different class of the ascomycetes that they forms the association with the lichen thallus, but also there are other lineages of the basidiomycetes. You could also see there are many basidiomycetes. They also, but in comparison to the ascomycete lineages, the basidiomycetes are relatively less. There are a few also that chytrids also has been found in a lichen thallus. Apart from this, algae that we know, the lecneromycete that we already know, and the other class that we have shown here. And the lichen also includes not only these. In a recent study that I supervised a PhD student, and we found that the lichens has also the sequences of the viruses. So we don't know whether the virus has any role in the lichen symbiosis, but they are present. In lichen thallus, we could find the main fungi that we call the lichenized fungi, other fungi that are belonging to different class of ascomycetes, basidiomycetes that belong to also the different lineages of basidiomycetes, chytridiomycetes, and viruses, non-photosynthetic bacteria. These are all present in the lichen thallus. Because of that, called a holobiont. And also to advance the knowledge of the lichen thallus, we have also found not only the one algal species are included in a lichen thallus, but is in a single thallus we can find more than one algal species forming association with lichenized fungi. So because of that, lichens are considered as holobiont. So this is the current hypothesis of the lichen symbiont that we can show you. So this was not my main target, but I just wanted to draw your attention on that. As you can see along the phylogenetic tree, the lichenized fungi not only grouped in Ascomycota, but also a few lineages are grouped here in Agaricomycota. This also form the main. I'm talking only the main lichenized fungi that forms the symbiosis. Some groups, most of belongs to the lecneromycetes, but also some are urosiomycetes, luteomycetes, also doxidiomycetes, soldariomycetes, and these are the lichenized fungi that do not form a monophyletic cluster, but they are polyphyletic. You see, polyphyletic means they do not group in a single cluster, but they are the lineages are spread all along the phylogenetic tree of the fungi, as you can see here. So lichenized fungi, as you all know, currently includes over 20,000 described species. These are the estimate of that we know. These are the species that we really know that there are 20,000 species. But if we go in the estimation or huge number of species that has been estimated in case of the lichenized fungi, including the fungi that are not lichenized fungi, there are more than the 2.5 million species that has been estimated so far. There's some estimate, they say could be like 3.5 million, but the current that we could trust on that could be like 2.5 million species that are estimated. But we only 80,000 species of the fungi in general that we know that has been described. As you can see, the lichens if we go in Escomicota, they represent a huge number of species. In Escomicota, there could be around 50,000 species that are described, 40,000, 40% species that could be like belongs to lichens. So lichens fungi represent a huge number of species in fungi. And other advantage of the lichens fungi, they are widespread. See, they are found in all the continents, including the extreme habitats like in Antarctica. They are also found in Antarctica. Here, this is Australia. This is also found in Antarctica. So all the continents, the species are spread. They do not confine to a single locality. It's even a single species can be found in all these continents. Example, I will go later. I will tell you later. For example, Parmelia saxatilis that are found species is found in all the continents. They are found in here, including Antarctica. So this is the only organism that are so such wide is the distribution comparison to any other living organism on the earth, on our planet, is the lichens that are shown, that has been shown, or so the wide spread distribution. So traditionally in lichens, the reproductive mode has been used to define the species. What is the reproductive mode? For example, the asexual or vegetative reproductive mode, that are the Acedia shown here. This is the propagules. They detach from the lichen thallus and spread through the wind or through the animal and colonize in a new habitat. Another is the Soridia. This is the powdery mass. This is also spread through the wind. It includes both the algal and the fungal partner. So both they spread and they have the ability to colonize a new habitat and form the complete thallus. And also, and the apothecia, this is here, it has been also used to define the species because this is the sexual reproductive structure, like in other fungi. They have the ascospores, and the ascospores have the ability to spread through the wind, but they need to find a compatible algae to form a lichen thallus. This has been frequently used in lichens to define a species. Apart from the vegetative structure, apart from the reproductive structure, the chemistry has also played important role in lichens to define the species, especially the medullary chemistry has been frequently used to distinguish species in lichens, but also the cortical chemistry has been used to define to segregate even genera in lichens. So the chemistry has also important roles in the lichens. So having in account what the features that has been used, are frequently used to define the lichens, I ask a question. Why an accurate sample identification, or we could say in other words, a species identification is needed to estimate lichen biodiversity? Why it is necessary? Why we really need to know this is the correct species or this is the correct sample? In other words, we should say this is the sample, this is the correct sample, and why it is necessary. Because in lichens, it is very important because the lichens present very few morphological features, not like other organisms, not like the higher plants has the more morphological features. Even the non-lichenized fungi also has some, not the microfungi, we could say. They have also the more morphological features, especially the productive features. They have the conidia or ascospores. They're very important in the defining the species. But in case of the lichen, these are the features that are very scarce. Even if they are present, most of them are highly plastic. I mean, they are not useful to define a species. That are these features, morphological features, do not form a monophyletic group. So they are spread in all along. This is the one region. Another, lichen species are frequently used in biomonitoring for air pollution, sensitive to air pollution due to their efficient absorption system and longevity. As we know, lichens can live for years, thousands of years. They can live on a substrate, and because of that, they are frequently used for biomonitoring species as a bioindicator for air pollution. So this is another region that we really need to know what is the species that we are talking about. Another region is that lichens, especially permeaceous, that we have worked so far in that. So the high pharmacological potential, such as antimicrobial, antioxidant, cytotoxic, anti-inflammatory, analgesic, and enzyme inhibitory actions. So this is another region that we really need to know what is the species, what is the sample that we are talking about, whether this is correct or not correct. Because later you need the industrial application, so it is also important. On top of all these, it has been frequently shown that the lichen includes the high number of cryptic species. Cryptic speciation is a common phenomenon in lichen. What is the cryptic species? The species that are morphologically similar but genetically distinct are the cryptic species, and this is very common, very frequent in lichens. Also in non-lichenized fungi, especially in microfungi, in Aspergillus penicillium, cryptic species is also common. But in lichens, the recent studies have shown that cryptic species are common in all the lichens. Not only, I will show you some examples, but mostly they are more common in macrolichens comparison to micro. Could be the macro lichens has not been studied using the molecular phylogeny so far. So because of that, we have more knowledge on the cryptic species presence in the macrolichens comparison to the micro. But I'm pretty sure when we will apply the phylogeny DNA sequences in macrolichens, this will be the same. So these are the regions that we really need to correctly identify the species to estimate the lichen biodiversity. So I ask another question before going on the topic. Are cryptic species common in a particular taxonomic groups? Let's say in a particular genus or particular family or particular order, or species, or they are common species with the different reproductive modes and distribution area? So this is the question that I asked. Whether the cryptic species is common in, let's say, sexually reproducing species, or they are more common in asexually reproducing species, or they are more common in the widely distributed species, that they have wide distribution compared to restricted species. All these, the question that I asked, and we'll show you some examples like that, and let's see what's happening. So I have outlined my talk in the different topics. I will show you some examples of species reproducing using the vegetative propagules, that is Soretia, and has cosmopolitan distribution, and also some examples vegetative propagules reproducing with Isidia, with also common cosmopolitan distribution. I will show you some examples also species with sexual reproduction. I have said in inverted commas cosmopolitan because they are not really cosmopolitan as in comparison to the asexual reproduction species. In the last, I will conclude the talk. So the first cryptic species in lichens was detected in 1997 in Parmelia sulcata. Parmelia sulcata, a species that reproduce asexually and has wide distribution. So there are the Crespo et al. In 1997 described, found that in studying the population of the 261 samples, gathering from the 6 countries of the 3 continents, and they found that in Parmelia saxatilis, there were the different ITS fragment lengths. I'm sorry. This is some ITS fragments. So they had 800 base pairs, and some had the 600 base pairs. So the authors reported at that time in 1997 that in sulcata has not only one species, but it may include other species, that the authors reported the different sequence length. And if you know, the ITS has the length of the ITS is 600 base pairs. So they found some that has 800 base pairs, and the differences was due to the presence of group 1 intron about on the three sides of the SSU, three end of the SSU, they found like about 200 base pair of group 1 intron. Due to that, they had like 800 base pair this, and the other has 600 base pair. Later, in a later study, I will show you the population that has the 800 base pairs was described as Parmelia incryptata, but the detection of this species was done in 1997. If you remember, the PCR, the technique PCR was invented in 1980s. So at that time, and the first species was described in after 17 years. Later, I could say, applying the molecular techniques in Lichen in 1997. And Parmelia sulcata, as I already told you, this reproduce with the Sorelia as a vegetative reproductive modes and has been described by Taylor in 1863. And it is also distributed in widespread in all the continent except Antarctica and Australia. This species is not distributed, but we could consider this is more widespread species. So the study done in 2005, yeah, the authors found one species within the sulcata complex. Yeah, they found monophyletic cluster, and the study was done with the two markers, with ITS and mitochondrial SSU. They do the phylogenetic tree. Using the phylogenetic tree, they reported that the sulcata is not monophyletic, but it is paraphyletic. And here, this cluster, they defined as a different species that is called Parmelia barinoi. It's dedicated to the one of the professors in Spain is named Eva Breno. And later, using revising morphological features, the authors found that this clade has the simple risings, whereas the Parmelia sulcata sensu stricto has the squarose rising. So it is defined with Soredia on the upper surface and squarose risings on the lower surface, and especially Parmelia as a genus, it has the epigorate pseudocephaly on the upper surface. These are some features that we found. Now, this Parmelia barinoi, in recent studies, we know that this is not only occur in Europe, but it's also found in North America and North Africa. So this is not so the restricted distribution either. In a later study in 2011 that I was telling you about the story of this, the group 1 intron, the authors fixed the taxonomy of the Parmelia sulcata. The species was described from Ireland. We went there and we collected the sample. We were pretty sure that the sensu stricto will cluster in one of the two clades, but unfortunately, they were distributed in both clade, in this clade and in this clade as well. The sample from the species. And one clade, this clade has the group 1 intron, all the samples included here, has 800 base pairs, and this has the 600 base pair. And this taxonomy was fixed, and we defined Parmelia sulcata, and the sample from Ireland is clustered here. And since this is the widespread species, in that case, we consider the widespread clade as sensu stricto, and that has the restricted distribution. We define a new species called Parmelia incryptata. Incryptata means that there is no morphological features, neither geographic feature to define this species or to segregate this species from Parmelia sulcata, because it is distributed in Spain, it is distributed in Ireland, from where the Parmelia sulcata sensu stricto was described by Taylor in 1836. So this was a species was detected in 1997 and formally described in 2011 as Another example that we are currently working on that. This is Parmelia, Parmotrema reticulatum. It is very common in India, as we all know. It is one of the widespread species in India, in Indian subcontinent. The species is distributed, defined with the Soridia, reproduced with the Soridia as asexual reproductive modes, and has the reticulate maculae on the upper surface, and using the, not only in India, it's also common in all the tropical species, is very common, and also it's common in Europe. And using the multi-locus data set, and not only the applying the genetic markers, but also applying the methodology, and we applied the coalescent-based species delimitation approach. And using the coalescent-based species delimitation approach, we found that in Parmotrema reticulatum is not only one species, but it may include around 10 cryptic species. These are all the lineages that we define that Parmelia reticulatum as currently defined includes about 10 cryptic species. We have not formally described it because it was very important to include the type of species. And the last year, I went to Ireland. Again, the species was described from Ireland, and we fortunately we collected a number of Parmelia reticulatum from that, especially from rock that was described. It is very important what I'm telling that once you found the species is polyphyletic, it is very important to define which of the clades belongs to the census structure. For that, we need a fresh material. We need to sequence the type material. Type material is impossible to sequence because that's very old. So we need to go to the type locality. We need to collect the material. We need to sequence and then to fix the taxonomy. And later we can define the species that are the clade.
Professor Pradeep, we can't hear you.
The sample didn't work locality to collect. So the paper will come soon that we are working on that. So we will formally describe all the species that are in reticulatum, all the 10 species, and let's see which of the clade is census structure. Till now, we really do not know which of the clades is the Parmotrema reticulatum census structure. You will see hopefully in this year. Paper will come out. So now to some examples of the CD8 I will show you the classic one. This is the Parmelia saxatilis, was described by Linnaeus in 1753. Saxatilis is a species that are defined having an CD8 upper surface, pseudocephali, figurate pseudocephali on the upper surface, and simple rises on the lower surface. It is quite distributed. See,
Divakar, the slide is of still Parmotrema reticulatum. Is it moving?
Oh, now?
No, same slide.
Oh.
Yeah, it is in okay.
Okay, now it's fine.
It is in the small screen. I mean, now it is saxatilis. Okay.
Now it's okay. Yeah. So the Parmelia saxatilis, that is, it's distributed in all the continent, including Antarctica, except Australia. In Australia, this is the one very similar species that is not saxatilis. It is also circumpolar, so the circumpolar distribution from Arctic to Antarctic region. That we call this is also in Arctic region, in let's say in Greenland is also there, in Antarctica is also there. So these are the current distribution. So let's see what happened in saxatilis. I will quickly go on that. So the molecular studies that was done in 2002, they defined Parmelia LNST based on the morphological feature. Parmelia LNST was segregated from the saxatilis, and in 2004, Parmelia serrana was segregated also from from Parmelia saxatilis. So these are the two, I would say, cryptic species, because you could see practically no morphological features has been detected. Even the author when described, they saw some morphological features, but after investigation, revising the different materials, we found there is no morphological features. It is still Parmelia saxatilis sensu stricto, so the bipolar distribution species, that is the distributed especially Serrana and Mediterranean region of Europe and North Africa, and ST in Western Europe is very common there in the temperate region. And
Yeah, again, some issue, Divakar. Probably network issue.
yeah,
Yeah,
Sanjeev?
we could not hear a few seconds
Oh,
before.
okay. Yeah. So, and authors using the two genes. But in North also very common, but especially in the low altitude region. This was in a region that we call this the cryo-complete ultra humid areas, was defined using the ecological terms. It was segregated, defined as Parmelia sulimei, and another is Parmelia imbricaria, using both the morphologically, both using the two gene markers, ATS and MCM7. It was segregated from Parmelia saxatilis. There has been segregated two more species. Let's see what's happening in 2020. So using the same markers in ITS and MCM7, so another lineage was discovered in Parmelia saxatilis sensu stricto, that is the Parmelia rajoy, was described from southern Spain. From here, this shows the restricted distribution in the Parmelia saxatilis. So let's see how many species so far. Yeah. Parmelia saxatilis sensulatum. So described Parmelia saxatilis in 1753. We never thought that Parmelia saxatilis is not a single species, but is a group of species, a complex of species when he was describing at that moment. Remember, without the molecular technologies, without the DNA sequences, without the molecular phylogeny, this would have not been possible to describe all these species, to segregate all these species, and clearly define how many species are currently included in Parmelia saxatilis. We really need to thanks to the new technology, to the PCR, and to the molecular invention of the molecular tools, that we really we now can know how many species are hidden in a single morphospecies. That is that I saw the example. This is the Parmelia saxatilis. On top of that, Parmelia saxatilis is still a wide species. Saxatilis sensu stricto, I'm telling, is still a wide spread species, and also circumpolar species is also included in both the polar Arctic and in Antarctic region. In Antarctica is the Parmelia saxatilis sensu stricto, that is there in the Antarctic Peninsula, that are the saxatilis sensu stricto, not other species are there. So this is really a classic example of the cryptic species. Another example, but just I will go quickly. This is Punctilia reducta. You know, Punctilia is defined using a punctate pseudocephaly on the upper surface and simple risings on the lower surface, and it has the acetia on the upper surface. In India, common.
Divakar, just minimize the slides and again show the slides.
Oh.
The slides are not moving.
Sorry.
Yeah, you can project this slide because I mean the previous slide also. Previous slide was
Yeah,
missed.
here, this one.
This one, yeah, this one. Okay. Now you go ahead to the next slide.
Yes, this one. There, you could. So,
Yeah.
you could, it's now we can show.
Yes, 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 yes.
Okay, okay. So punctilla reducta is it's very common in India. So using multi-locus data set and applying the coalescent-based approach, we found we fixed first we fixed what is the punctilla reducta syncyticto. This is the clade. Reducta is sorry. This is the syncyticto that is described. The reducta is described from North America, and the samples were tested here. And this is the lineages that we from Canary Island, and we described one species. This is Juan. And then the samples that was distributed in Asia is defined as ruderata. So now in India, the reducta is not reducta, but the reducta that was described and that has been reported from India is ruderata. The reducta is only restricted to North America. And the African lineage was also defined to the different species, but is not formally described yet. So now let's come to the sexually example of the sexual reproducing species. So this is the one species that I'm showing you. It Parmelia quercina. This is not distributed in, not known in Asia. It is sorry, not in India. It is segregated using the apothecia on the upper surface. And simple cilia on the lower surface, and also the simple cilia in the axials, and simple rhizoids on the lower surface. It is distributed in North America and mostly in Europe and in Australia. Because of that, I put in inverted comma, that is not as common a species as a cosmopolitan species, as I saw the examples of the 38 one. Let's see what's happening here. Using the 3 gene data set, authors define that in Quercina, there are 1, 2, 3, 4 or 5 lineages they found. 5 lineages in Quercina, the authors have defined, and then they also used morphological features. They found some morphological features, especially in maculate surface using the transmission electron microscopy. The authors found some differences on the upper surface, and also on the ascospores there were some differences. I do not want to go in detail, but I only want to underline that one specimen is sufficient to distinguish Quercina and Carporisens using the DNA sequences. However, this is not the case of the Quercina and Colei. We cannot define with this. This is difference between Carporisens and Quercina. Colei and Quercina was impossible to define. And the Australian lineages was described as a new species as Parmelina elixia. So Parmelina carporisens, as has been described initially, is now include, let's say here, the 3 or 4, 5 species, and the Quercina syncyticto is here distributed to the Europe, is restricted to the Europe, and this is the Carporisens is also to the Europe, and this is the North American lineages is Colei, and the Australian lineages is Parmelina elixia, and this is now defined as is shifted to a new genus. This is the Austroparmelina. What I'm telling to say, Parmelina quercina is not only included the different species, but a different a new genus was hidden in Parmelia quercina morphological concept that was using this. We could see this is Parmelina syncyticto. You could see here this is the Austroparmelina. This includes the Parmelina quercina that was described using the morphological species. It also includes other species of Parmelina that was described from Australia, and there's no Parmelina in Australia. That all the Parmelina in Australia belong to Austroparmelina. So let's come with another example. This is the Melanelexia glabra. This is another species. This is also distributed in India. This is a foliose thallus with brown upper surface, apothecia on the upper surface, and cortical hairs. And they call fenestrate pores on the epicortex. It shows the disjunct distribution in Eurasia and North America. This is not a cosmopolitan species. We see when we applied this with molecular phylogeny and in the DNA sequences, we found this is a clade that is distributed in the North America, and this is the Eurasian one. So this clade later was described as a new species, that is the Melanelexia californica, because this is only restricted to California. Later on, there was another clade here. This is distributed in Europe, in Anatolia, especially in Turkey and in that region. And this species was, this clade has been raised to a name has been opted for this, and this is Melanelexia epilosa. So now in Melanelexia glabra is not only one species, but is included 3 species we can find in Melanelexia glabra sensitive. This is the epilosa. This is in Europe and in Turkey, and I don't know this from India was, this is Spain, Turkey, and only in Spain and Turkey is epilosa is distributed at the moment. So as you can see, this is the Californica and this is the glabra. One specimen is not sufficient to distinguish both species morphologically, while a single sequence is more than sufficient to distinguish the Melanelexia glabra from the Melanelexia californica. In this case, the geographical features play important role. This is restricted distribution to California, but this is not the case in all the cryptic species. Especially in this, the geography, we know the glabra that is distributed in California is californica, and that is distributed in Europe and Eurasia is Melanelexia glabra. And in 2016, there are a number of species, cryptic species has been defined around family line. Let's say in Melanelexia, that has been later I put in red color. This has been described a number of species in Melanohalia. There has been described a number of species based on the DNA sequence. In Montanellia also, there has been described in 2 species. Remember, Montanellia, the genus Montanellia was defined in 2012. I described 2012 the genus Montanellia based on the molecular phylogeny and the morphological features. Earlier it was grouped in the Melanellia sense. Stick to Melanellia, that was Melanellia, do not belong to familioid lichen, but now Melanellia belongs to the Citeroid class. Because of that, I'm not telling about this, the Melanellia. So this was the case. I do not want to go in deep. So now it's time to conclude the talk. So as I have repeatedly shown in my talk, so this is the appearance of the cryptic species is a common phenomenon in macrolichen species. As I was telling in the beginning, that the macrolichen, I'm telling the macrolichen species because macrolichen species has been more studied applying the molecular tools than the microlichens. But this could be also the case in macrolichens as well. But with the data at hand, we could say that the cryptic species is a common phenomenon in microlichens. And a high number of over 150 cryptic species have been detected in macrolichen single family, that is Palmoliaceae. So far in Palmoliaceae, we have detected more than 150 species. Even not all the species has been formally described, but has been detected over 150 species. That could be like the 200 or 20% of the sequential species of the Palmoliaceae. I didn't say Palmoliaceae include around 3,000 species. That those who are not lichenologists, just to let them know And we have only sequenced 20%. We have only studied the 20% of the total number of species of the Pomeliaceae. So sexually and asexually, that I asked the question. Remember, are the cryptic species is common in a particular taxonomic group or in particular reproductive modes? So let's say here is the answer. Sexually and asexually reproducing species distributed widespread cosmopolitan species may include several cryptic species. I'd ask question, there are two, whether the cryptic species are more common in sexual species, cosmopolitan species, species with distributed, showing the restricted distribution. These are the questions in the beginning that I asked. So as I have shown the number of examples, sexually and asexually reproducing cosmopolitan species may include several cryptic lineages. So they are not restricted to the reproductive mode. They're neither restricted to the reproductive mode, neither to the geographic region. So you can find the species that are cosmopolitan, and that's also the restricted distribution. Lichen species, especially sexual, have more restricted distribution than previously assumed. Using the molecular data, we confirmed that in lichen species that reproduce sexually shows the more restricted distribution. In case of cholei, let's say distributed to North America, California reproducing sexually distributed to the North America as well. But if we go in the asexually reproducing cryptic species, let's say Parmelia serrana is also widespread. Paranoia is distributed in North America, but this is not the case of in the sexual reproducing species. They show the more restricted distribution. So new cryptic lineages were found in each and every macrolichen species that have been studied at population level so far. So far, whatever the number of species that we have studied at population level, we detected the cryptic species. So ending the conclusion, so DNA sequence-based sample identification is necessary for species circumscription in lichens, not only in lichens, in especially microfungi also. It is crucial. Using the DNA sequence is very important, or I will say is mandatory nowadays to circumscribe species. Thus, this is needed to estimate the lichen species biodiversity. Not only the DNA sequences is necessary, but also the bioinformatic tools is also really important to circumscribe the species. Multi-locus sequence data should be analyzed both in the phylogenetic and the coalescent-based framework. This is very important also to circumscribe the species in lichen. So in nowadays, just using the morphological features is not enough to estimate the lichen species biodiversity, because we cannot estimate using morphological features. It is impossible to estimate the cryptic species that are hidden in a number of widespread species. So for lichen species biodiversity estimation, it is necessary to use the DNA sequences, and second, using not only just ITS, because using the ITS you could create a phylogeny of a gene but not a species. At least the two gene is needed to correctly circumscribe a species. So to estimate the number of species, to number of the lichen biodiversity, it is necessary applying multi-locus data set and analyzing them in coalescent-based species to show or to explore the hidden diversity in lichen. So what we are working now in this case, So this is that we know the species are present there. So these are typical species in one, in another one. But what are the factors driving the cryptic species and in lichen? So this is the another question that we really need to know. What are the factors that drive the cryptic species? And for that, so we are currently working on that. So we have a project going on genomics. So this was our the project. We applied the metagenomics tools for characterizing biodiversity in lineages of symbiotic organisms rich in cryptic species that we choose in Pharmacognosy because the Pharmacognosy is rich in the cryptic species, and then we will evaluate the evolutionary process that generate this diversity. So this the project that is currently going on. So this, and we have another project in parallel that we will apply also using both the genomics and bioinformatics tools to really understand what are the factors that are driving cryptic species and in lichen. This is very important. So on this, I will shut my mouth and thank you all for listening me, and I'm now ready for answering the question. Thanks a lot.
Oh, thank you, Professor Pradeep Divakar, for that wonderful presentation. Can you hear me, Professor Divakar?
Yes, I'm hearing you.
Maybe you can stop presenting so that we can have a
Yes.
better visuals. Okay, thank you. Thank you very much for talking about my favorite topic about speciation. So, I mean, I had worked on coltotrichum, siamens. Then we found that there could be 7 cryptic species. But when the coalescent method was applied, it was found there are not 7 species. There is only 1 species.
Yeah.
So my point is that GCPS are becoming a tool for splitting and coalescent becoming a tool for clumping. What is it like now? At least from your experience from the lichenology.
Yeah, from my experience from lichen point of view, I mean lichen and fungi that we have worked so far on that. So it is very important for us to have the species tree. The species tree is very important because if you have the gene tree, you are not really having a species tree. So for a species tree, it is very important to have the coalescent-based approach. So I trust more what is telling the coalescent-based approach to define a species. If it's saying just it's one species, we have to trust more than having a gene tree. So gene tree, because in gene tree we cannot, it could be like incomplete linear sorting. We cannot resolve the problem with the gene tree. Paralogs is also another issue in general in lichens or in fungi. So it could be due to the paralogs, you are having the different species using the gene tree. But if you put all in a coalescent-based approach, you could resolve all these issues. Especially this is many programs we now apply this PPNP. That is also a very good program that Astra is also using. So PPNP we use both for defining the species using the coalescent-based species and then validating. But most important, the definition. First, we need to define and then to validate whether these species are validated with the coalescent-based approach. So my recommendation will be applying coalescent-based approach and trusting the results on coalescent-based approach.
Thank you for that answer, Professor Divakar. My supplementary question would be, even coalescent method would yield in speculation or a hypothesis about the genetic barrier. So to confirm that, we need to do mating test. At least in the case of coltotrichum, at least researchers in coltotrichum taxonomy did the mating test and proved that there were not genetic barriers. Those were the same one species. So do you have a similar approach in lichenology also?
Sorry, I'm not hearing. There is a noise somewhere. Yeah, could you repeat again the question, please?
So what I was trying to tell is even if you employ coalescent methods, that would not give you confirmation. That would just generate hypotheses about the possible genetic barriers among a set of tags. Yeah. So to confirm it, we need to do mating test. At least in case of Coltotrichum, researchers did mating test and proved that there were not genetic barriers. That because of that, this is a single species, not 7 species as previously described. Do you have similar approach in lichenology?
In lichens, yeah, we do have this. I mean, it is very difficult to do the crossing with in lichens, with the mating system. But what we do in lichens, we study at a population level and see whether there is allele sharing. If there is allele sharing, I mean, the alleles are shared, we cannot define the species. So alleles should be fixed in all the clades. So this approach that we use to validate the hypotheses that are given with coalescent-based approach, and there is some program, as I said, BPNP is also used to validating, not only to segregating, but it's also validating your species that has been detected using the genetic approach with the coalescent-based approach. But it's very difficult, with culturing and then to saying whether this is the mating, different mating types, single cell culture is not difficult in case of lichen fungi. So it is, even we could sequence the mating type genes. And in that we have primers. We define the primers for mating type genes. We study the mating types in lichens using genomics. So the paper was published in 2019 on that. So we could do that. Could be in one of the papers we published in scientific report was that at population level, we really interested to know what are the mating types that selects naturally, that the nature is selecting. So we studied more than 500 samples from the different continents. There were 2 clear populations, but we found the answer was that 50% population had mating type 1 gene and 50% had the mating type 2. So there was not any difference in the selection of the mating genes. So this could be applied, but it's not helpful to validating in case of lichen. Yeah.
Thank you, Professor Pradeep Divakar. Ladies and gentlemen, if you have any questions, please use the option raise hand in Google Meet. Dr. Sanjeev Naik, you have any questions?
Not really, but still, Dr. Divakar, I mean, these cryptic species, they are specifically, I mean, defined to a geographical area. I mean, certainly they are in the way of becoming a new species or getting distinctly a new species. I mean, how long it would take for a new species to be created out of from lichens? What is the speciation time in lichens?
Sanjeev, in species, for knowing the species and time, in some of the papers, let's say I saw you in Punctilla reducta, and also in Palmoptera reticulatum, no, but in Punctilla reducta, we did the molecular dating. And with the molecular dating, and also I didn't show the examples in Brioria, Fuchsescens complex, we also did the molecular dating. With the molecular dating, you could really know the species and time of the cryptic species when the lineage was separated from the sister species, from sibling species, and in some cases it could be like they're very recent. They're very lichens. They're very comparing to higher plants. They are still old, but they are very recent. Could be 1 million or 2 million years old. The species that we have defined with the cryptic species. So applying the molecular dating, you could really know the species and type of the cryptic species.
Yeah, I mean, second thing is that cryptic species are found more in macro lichens. Is it because that macro lichens, since they are large lichens, so easy to handle and extract DNA and all? Since the macro lichens are studied better, that is why we see more cryptic species in macro lichens than the micro lichens. Is it so, or is there any other reason?
I think there's two reasons. One is that the microlichens is more studied. I mean, both from morphological point of view and molecular point of view. Because of that, we have more knowledge on that. There are more cryptic species in microlichens comparing to microlichens. In case of microlichens, I know example Graphis scripta that includes, I think, 6 or 7 cryptic species in Graphis scripta as well. So could be that the microlichens is less studied, applying the molecular techniques, because it is not really easy to sequence, to isolate the DNA or from the microlichens, and especially to sequence, because there are other fungi that they are hidden and the sequencing is not really easy in case of microlichens. This could be one reason. Another reason could be because in microlichens, when we have defined the species, and the morphological features that has been used was not so, I mean, I would say that we have not been very careful when using the morphological features for defining the microlichen, because you could really see these are the features, Zedia, Sorelia, chemistry, and we define the species. So this could be also other reason. In case of microlichen, there are other features that we go in deep. We need to measure the ascospores, conidia, and all the anatomical features. So because of that, could be that they are well defined comparing to microlichen. So yeah.
Okay, thank you. Others may ask questions, please.
Dr. Nagendra Chandravanshi, please.
Yeah.
Dr. Nagendra, can you hear us?
Yeah. Hello, sir.
Please ask your question. Thank you.
No, sir, Pradeep, sir. A very nice session. Sir, I am actually I am working in mushroom. So most of the time I found some lichens on the mushroom body. So,
Yeah.
so how can we characterize and the relationship between microbiota and the phycobiota, sir? I'm just a simple query.
Sorry, could you repeat the question? It's echo is coming.
Yeah, sir, mushrooms already having some time, most of the time, mushroom also associate with these lichens in the field, like a forest, like a wood sample.
Yeah.
So we can some confuse. They are a symbiotic mechanism. They are associated. Yeah, there are some other difference mechanism like they're protecting your selfing, your food source.
I mean, for knowing this is really symbiotic association, we really need to know there is an exchange between the two organisms. So most of the mushrooms that we know that they are the symbiotic, the fungi. They form the symbiosis with the root of the high plants. So they are not just attached, but they form the association with the root of the tree. We know the mushrooms. I mean, in India there are also many, but in Europe there are quite a number of, and there are few species that are particularly common in a particular forest, that they can grow in pine forest only below the pine tree. Some are below the quercus tree. They grow these mushrooms. Because of that, they form the symbiotic association with the root of those plants. Yeah. That was your question? Yeah.
Yeah. Okay, sir. Sir, got it, sir.
Yeah.
Thank you. Ladies and gentlemen, if you have any more questions, please raise your hand using the raise hand button in the Google Meet. Looks like there are not any more questions. Dr. Sanjeeva, maybe we can close the session now.
Yeah, thank you, Damodar. And yeah, on behalf of AFB and ILS, I thank Professor Pradeep Kumar Divakar for giving a very nice presentation, a very nice overview of estimating lichen diversity, where considering the cryptic species is very much necessary, and molecule tools are very much needed to be applied for our routine work. So that is how we will be able to successfully estimate the diversity of the lichens. It was very enlightening talk. I thank Dr. Divakar for sparing his time for this talk and for this series. Thank you, Divakar.
Thank you, Sanjeev. Thank you for inviting me. It has been a very great pleasure to me to be here to show my knowledge to all the colleagues of the both societies, and I'm very, very happy for that. Thank you. Thanks to.
Yeah.
Thank you, Professor Divakar. I mean, it has been wonderful listening to you. I mean, I am a big fan of you. I remember reading your 1990s paper when I started my PhD in 2003 at the University of Hong Kong. I think the 1991 paper, I think, where you used the ideas. It has been a pleasure listening to you. Thank you, Professor Divakar, and we would like to see you again on this platform. Some maybe
Sure.
some of the summer. Yeah,
Yeah.
thank you.
Okay. Thank you. Thank you.
Thank you. Thank you. And I would like to thank the Indian Lichenological Society, their members, their president, secretary, treasurer, everybody for collaborating in the Association of Fungal Biologists. I would like to thank the esteemed members of the Association of Fungal Biology, the president, Dr. Sunil Kumar Deshmukh, the secretary, Dr. Shashireka, treasurer, Dr. Mahavir Gosavi, and the other members of the managing committee and the other life members and the members of the association for your active participation. And today's audience, you have been great. I wasn't expecting more than 50 people. I could see 80 plus people. So, thank you very much for your kind participation. With this, we sign off. Thank you, Dr. Sanjeeva. Thank you very much.
Thank you very much. Thank you all the audience. Thank you, Dr.
Okay.
Belle.
Yeah, bye-bye. Have a nice Sunday. Bye-bye.